All right. So this, and yeah.
If you can sign this afterwards.
Sure. I'm hierarchically challenged for the microphone. Okay. All right. So this is what I thought I can talk about. I can talk a bit about flight development and stochasticity in flight development because I got the feeling that most of you care more about signaling networks and things like that. Okay. So this is plan A, flight development and real data. Scary. And then a bit of abstract stuff about information theory and dissipation. This is plan B, and I'm also showing it in case we go with plan A, but you still are interested in plan A. A lot of what I try to understand is immunity. So one of the things that we've done is we've tried to show why there's a diversity of different immune systems in the world, in different organisms, from Christmas to adaptive and innate systems, and come up with evolutionary arguments for that. So it's also stochastic, but not in the sense of stochastic networks or anything. Another part is sort of trying to understand within the adaptive immune system why we have a diversity of receptors. We get these power law distributions of receptors, and we can explain that in terms of fluctuating fitnesses. So am I right that I should go along with flies? Okay, so let's go with flies. Okay. So yeah, so the fly is of course one of the model organisms for development. And what happens in early fly development is that the fly mother lays some bicoid mRNA here, then diffuses and turns to proteins and forms this bicoid, exponentially decaying bicoid gradient, which then regulates a bunch of downstream genes called gap genes, including hunchback. And already the gap genes have very precise boundaries, meaning they're expressed in some parts of the embryo and not in others. So this is hunchback expression, which has this very precise boundary, and then others have different boundaries, and together these form stripes which determine where different body segments will go, and how basically it's a blueprint for the development of the body. But as you've probably heard ad nauseum, is we're always surprised by the precision in development and how precisely this boundary is established and how is it implemented molecularly. And you're probably thinking right now, well, you've been hearing this talk for 20 years or however long you've been going to talks, and it's always the same talk, and how come they haven't solved it yet? Okay, and so I'm going to try and show you, I mean, the answer is they haven't solved it yet, and I'm going to try and show you a different angle to this very, very old problem. Just basically, how do you then get nuclei in this part of the embryo that turn on, and then in this part that they don't turn on, and you get this transition zone, which is a very thin boundary. And we're really trying to understand how it is implemented molecularly. So mine and my collaborators, which I think now that we're talking about it, I should probably go back and mention. So this is work done in collaboration with Nathalie Dostatny, who's a fly geneticist at the Curie Institute, and Mathieu Coppet, who's an experimental biophysicist. And most of it, what I'll talk about, was done by Teresa Ferraro and Tanguy Luca and Jonathan Depont on the theory side. Okay, and then when I get to the information theory work, it's mainly work done by Francesca Mancini in collaboration with Matteo Marsili and Chris Wiggins, and later we've been joined by Dario Villamaina and Paulina Szymańska. Okay, so back to this thing. So basically, what you usually see is a static picture, but in fact, when you start thinking about when it happens, the bicoid gradient is established around cell cycle 7-8, which is about 1 hour into development. And most of what the images you've probably seen are from cycle 14, but basically by around cycle 11, 12, the precise boundary is there. And this is all of these cell cycles together about 30 minutes. Each of these cell cycles, 9, 10, 11, is very short. It's really of the order of 5 minutes, right? As they get longer as you go on, but you know, a number to have in your mind is 5 minutes. Here we get to maybe 10, 15. But it's a short time. So as I said, in the picture we mainly have in mind comes from static picture from fish experiments. So to do a fish experiment, you take the embryo and you fix it, which means you kill it. And then you've tagged your mRNA with a fluorescent promoter, and then you see where a transcription occurs. And so you get these kinds of pictures from the different cell cycles, where the green dot means that there's expression at 2 loci, blue dot that is expression at 1 loci, and white that none. And you do see that the boundary gets pretty well determined, and when you average over different embryos, you see that the position of the boundary is determined within of the order of 8 or 10%. So it's a very well established boundary very early on, already from around cell cycle 10, that doesn't seem to change with the cycle, at least when you look at variability from embryo to embryo. But when you look at any one region, there is still quite a lot of variability. So you can ask whether the sources of variability, and I think this is something that a lot of you have thought a lot about, so I don't need to dwell on it, that when you see variability in a static picture, that it could be variability from the fact that many cells are doing many different things, and you have many cells expressing at different levels, or you could have that a situation where each cell or nucleus is actually expressing variably. So, and when you look at the static picture, you get the same kind of picture. So how can you address that? Well, one way is to look at the dynamics, and so to perform live imaging. So this is what Natalie's lab has developed, is basically a way of doing live imaging and tracing the expression from the transcription factor binding. Sorry, my throat is killing me, so I'm going to be coughing all for the next 30 minutes or so. Okay, not okay. Anyway, the way this works, it's called MS2 MCP. So if you like yeast, you're probably very familiar with this. What it is just you build in some mRNA into your gene of interest, and when this mRNA is expressed, it forms these MS2 loops, which then bind MCP GFP proteins, which light up. Basically, once you express the mRNA at the locus, you start seeing a signal. And so the question we're interested in is basically what does transcription really look like, whether your gene is non-bursty. Oh my God. Okay. Whether your gene is non-bursty and the polymerase is basically transcribing all the time at the maximum possible level, or whether the gene is bursty and there's periods of activity and not activity. The problem is that, as I said, so the first part of this talk is going to be about a theorist getting close to data. So the first thing you realize is that depending on where they actually put these MS2 loops, you're going to get a different signal. So if you put it before the gene in the 5' end, well, you're going to start transcribing, and you're going to transcribe the let's call it the fluorescent part first. So you're going to get a very high signal, and you're going to keep having the fluorescent loops at the locus. I'm sorry. So you're gonna, okay, if it's at the beginning, you're going to get the fluorescent loops, and they're gonna stay at the locus, they're gonna be bound close to the locus for the whole duration of when the gene is expressed, so you're gonna get a very high signal. However, if your reporter gene is at the end, then you're gonna transcribe your whole gene without anything happening, and you're only gonna get a weak signal at the end. But in this, so this may make you think that, okay, so I want to get a stronger signal because background fluorescence is a problem. So let's do that. But there's another thing, and so it's shown here on the example with specifically looking at fluctuations in the gene expression state. In the case we considered a cycle model because it's been shown in many eukaryotic systems that transcription happens in a form of a cycle where you have an on state and then many off states before you actually can go off. So let's imagine such a system, just simply because it's more general than a two-state system. And then we have a fluctuating gene, and we can consider systems where we have fast fluctuations and slow fluctuations. So in the case of the 5' where we put the promoter in the 5', so that's before the gene, we do get a very strong signal. The y-axis here is much larger than here compared to when we put it after the gene. But basically, the gene also buffers. It doesn't really matter when you have very rare fluctuations, but when you have very common fluctuations, and this is especially seen here, it's basically hard to distinguish noise from real off events. And this is made very clear when you look at correlation functions. So you look at the autocorrelation function of the fluorescent signal as a function of time for the case where the gene, when the reporter is after the gene and before the gene, and when it's after the gene, thank you. All right, thank you. You're actually able to capture the promoter fluctuations in the case when they're both rare and frequent. So basically you can, if it's going really, really fast, you're over here, you're going slowly. Whereas if it's after the gene, you're fine when things are slowly because essentially not much is happening. But when the promoter fluctuations are very fast, frequent, you don't have any detection power, and if your gene, and it actually depends on the length of your gene, if your gene is long, then you lose even more detection power. All the signal falls onto one graph. So just in summary, if you're trying to learn something about the fluctuations, the relaxation time, if you have a slowly switching promoter, you're essentially good no matter what, and then you can go for trying to avoid the background signal. But if your promoter is fast switching, you really do want to be able to track these fluctuations. So with that in mind, I'll let you watch the movie while I cough. They put in the promoter closer to the beginning of the gene, so closer to the 3' end, and here you see the developing fly embryo with the spots appearing in the anterior part, so the head part of the embryo, and not so much in the end part. Okay, so then this is what the traces look like. The X position is a function of the AP axis. This axis is minutes. This is the activity, and these are different nuclei. So you see, essentially, just by looking by eye, you see that this is a somewhat bursty promoter, which was not what was believed before. Yeah, I think I'll go for the cough drop. So, I mean, there's things we can learn from this. I won't go into terrible details. But a big question in all of this is, what does Bicoid actually do? So one thing is that we can look at different properties, and for example, so Bicoid, remember, is the gradient that activates Hunchback. So we can look at the initiation time cycle by cycle as a function of the AP axis. So the AP axis is basically the x-axis of the fly. It stands for anterior posterior, and the Bicoid, Bicoid is a gradient that decays along this axis. So looking at something as a function of the AP axis basically is looking at something as a function of Bicoid concentration. And you see that the initiation time actually completely does not depend on Bicoid. However, Bicoid does increase the duration of anterior activity periods, which can be seen here. So what you see here is a comparison of the sort of wild type, which is called MS2, in darker colors, to a Bicoid mutant where there is no Bicoid at all, and it's divided into the anterior and posterior region. So this is the only one that has Bicoid, and you see that this is needed to have a peak as a function of time at the later cell cycles. The posterior behavior, so there where Bicoid is low, behaves the same as the mutant where there is no Bicoid. Basically, there is much less expression as in the later cell cycles. Although, interestingly, in the beginning, there is some expression, and it's not that there is no expression. So actually, Bicoid is not essential to start Hunchback up. Hunchback will be expressed even in the negative mutant, but it is essential for then increasing expression in this head part, in the anterior end, and for, well, and basically differentiating between the anterior and the posterior. And it is essential for boundary formation. So if we again look at it as a function of this x-axis, in the beginning there is no difference between the mutant and the non-mutant, and then the difference starts to appear. This is the wild type, this is the mutant. So basically things will start going without Bicoid, but to get this precise boundary by cycle 12, or to get a boundary by cycle 12, you need Bicoid. So you could maybe think that, well, maybe if we could wait around until cycle 28 or 36, the boundary would still appear by itself. Maybe there's other factors going on. Maybe what Bicoid really does in these early cell cycles is something to do with timing. Because this is development, and we don't have all day, right? I mean, we literally have 30 minutes. Or the fly has 30 minutes. So maybe in all of this, timing is important. So in order to understand that a bit more, we wanted to look at the data in more detail. And so as physicists, we turn to the correlation functions. And this is really where we get into the real data, real problems business. So one limitation, well, as I said, time is a limitation for the fly. It turns out to be a big limitation for us. So this is the autocorrelation function of the fluorescent signal coming from this Hunchback promoter as a function of time for cell cycle 12 in black, 13 in, what is it, purple, and 11 in red. And the first thing you may notice is that they all have this dip that goes below 0, which essentially as physicists we don't see in correlation functions, right? Correlation functions should go to 0. And so usually when you see that, this is a signature of something interesting going on. Well, in this case, it happens to be a complete artifact of the fact that the time trace is very short. Right, and you can see the same thing in an Ornstein-Uhlenberg process. And so usually when we have a too short trace, well, we just go and take more data. But here we have a natural limitation of the system, which is that the fly doesn't have more time to take more data. It has 5 minutes and that's all we're getting, and then the cell cycle shuts itself off, and that's it. So so we somehow have to deal with it. The second thing is that if you look at this, you can see that there is a difference between the different cell cycles, and this also turns out to be a complete artifact of the short duration, that if you downsample the longest data set we have, which is cell cycle 12, we actually have more data for cell cycle 12 than for 13, because then there are some aggregates that appear in the data, so they stop recording. But if you downsample it, you basically see, this is not the plot that actually shows it, you actually see that all the cell cycles, then if you take the same number of points, which is on the order of 24, they all fall on the same line. So again, the differences you're seeing are pure artifacts of the finite traces. So what that tells us is that in any analysis we have to take into account that we have these very short time traces, and there's no way around it, because we have them, but so does the fly when it's trying to make a decision, any kind of readout. So what we did is we basically calculated custom-made autocorrelation functions for the system, where we have the setup, where we have the fluorescent reporter in front of or after the gene. It produces a fluorescent signal, which we can calculate using, so we assume a model, and we actually assumed three kinds of models: a simple telegraph model, a two-state model where the gene is turned on and off, a cycle model, because we, as I explained before, we had reasons to believe that this is a sort of more natural form for the dynamics, and then a model where basically where the rate for going back off is chosen from a gamma distribution, because this is a way of mimicking the effect of having many off states without actually having to have many off states. And so for each of these cases, we can analytically calculate an autocorrelation function and a cross-correlation function. And so this is just a comparison of the simulations to the analytical calculations to show that it works for the case where you put it in the 3' end, the 5' end, and somewhere in the middle, and for the different kinds of models. And so I'm showing you the result for the simplest case. I think I'm actually showing you even the steady state version, or maybe sorry, no, not the steady state version. So we can also do these calculations when we're not in steady state, because there's no reason to believe we are in steady state with the system. So we can. So I'm showing you the result for the two-state model that is out of steady state, and I'm not showing you the corrections, the explicit corrections for taking into account the finite trace, but we include that. So we basically, there's spurious correlations that are introduced by the fact that you have into the mean and the variance, and you can calculate them and subtract them. Okay. So when we do that, we can actually go back and we can try and see which kind of what kind of model best describes what is going on in the fly, and we can start with the anterior of the embryo. So this is actually the data, the red dots here with the standard error around them, and this is our best fit. It's actually, there's 3 lines overlaying each other. The red, which is the best two-state fit, the best two-state out of steady state, and the best free state. And basically we cannot distinguish them. Okay, what the green line is another hypothesis that was put forward, is that basically it's just Poisson firing of the gene. That there's no off state, but you're randomly turning it on. So that we can exclude. But other than that, these are all equally good fits. And since we cannot, so okay. So here I should say something else, because you can say, well, there's a simpler thing we could have done. We could have just looked at the distribution of the off times, and there we can distinguish a two-state from a cycle model easily. But again, given our finite traces, we don't have enough off-state statistics to really distinguish them. So we cannot do that. So in this case, we're really not in a position to distinguish the two-state and the cycle model. However, in the anterior end of the embryo here, bicoid is very high. Bicoid is so high that basically the gene should be constantly on. It should be full activation. So even if we have a cycle model, we should be in a limit that once you find yourself in state of 2, you're automatically put into on 1, because there's such a high activator concentration that this is an automatic reaction. So a cycle model should reduce naturally to a free cycle model should reduce naturally to a two-state model. A two-state model should reduce then to this Poisson firing rate that you're just seeing it. So then we can say, okay, so in that case, in the anterior rate, in the anterior part of the embryo, we're actually sure we have a cycle, that there are some hidden off states, because if there wasn't, then the Poisson firing rate would work. So that's what we think. So we say, okay, so it's probably a free state model. But then that argument, following that argument, saying that it's the same gene at the boundary, then we should be able to actually clearly see the free states, and we shouldn't be in this regime. But we're not. We still cannot distinguish. This two-state still works as well as before. Okay. But so for that reason, going with simplicity, we stick to the two-state model, and we actually perform, we use the autocorrelation function to learn the k-on and k-off states, and we have now done it by dividing it by in the anterior end and in the boundary. So the anterior are the blue points and the boundary are the red points, and the different signs are different cell cycles. So there's some variability, but you can't really see a trend with the different cell cycles. The cell cycles seem quite similar, but there is a clear trend that distinguishes the boundary from the anterior end. And in the anterior, you have a very high k-on, as you could imagine, because you have this bicoid that's basically telling you to switch on all the time. But in the boundary, you have a much higher k-off than in the anterior. So playing with k-off, of course, it's very weird to have k-off changing. And what we think is happening here is a cooperative effect, that once you have that you basically have a low k-off in the anterior, because when you have all these bicoid molecules bound to the promoter, it stabilizes the expression state and then decreases k-off. But you have these very different situations in the anterior and in the boundary. Okay, so getting back to the original question, well, what does this mean for precision? So we can go back and perform a sort of also rather traditional back-of-the-envelope calculation, but taking into account this new data. So the basic idea is, what is precision? Is the fact of how well can you measure concentration. And what a gene does, what a promoter does, is it's measuring concentration and figuring out whether it should be on or off. So basically, what are the elements you have? Well, you have the time you're actually performing the measurement, which is the time of the cell cycle. In the early cell cycles, it's 5 minutes. In the later cell cycles, like 13, it's maybe of the order of 10, 15. We put in the exact number that the trace is actually significantly non-zero into the calculation. And then there's another time scale of the problem, which is the time that it takes the system to go through one of these cycles, which is 1 over k on plus k off, which we can calculate from the inference we did. And so then the independent number of measurements of the concentrations that the gene makes is the duration of the cell cycle divided by how long it takes to go through one cell cycle. And then you have 6 copies of the gene, because you have, of the promoter. You have 6 promoters in the in each cell. So you have 6 times Tc over tau independent measurements. So then the error you're making in measuring p-on is basically your bimodal sampling error of p-on divided by the number of independent measurements. Okay? So when we look at so theory is basically this argument with the k-on and k-off inferred, as I showed you on the previous slide. And we can compare this theory in the anterior and boundary at cell cycle 14 to a direct measuring from the data. So you take the embryo, you divide it into the anterior and the boundary region, and you calculate the mean p-on in each nucleus. You average that, and you then calculate the variance in the p-on over the nuclei. Okay? So you basically see that in the anterior, I mean, they agree, I would say, given, you know, the crudeness of the measurement. Here we have of the order of 30, right? Let's say 28. Here we have of the order of 15% accuracy. So the first thing, so one thing is that these 2 independent ways of calculating it seem to agree in cell cycle 13. I'll give you a caveat that in the early cycles they actually don't agree so well, and we think this is because there's sort of calibration problems with how we measured the p-on directly from the data. The other thing is you clearly see that in the anterior region you have pretty good precision of the order of 15%. In the boundary region you're at 30%. So that's not great. And then if you look at the theoretical predictions, which I say we trust more because of the of less problems with calibration, it basically averages itself out a bit more. You see that the precision increases with cell cycle, both in the anterior and in the boundary, but remains sort of too low in the boundary. So we have this more than 10%, and if we wanted to get 10% from all of this, we would need to average for 45 minutes. So we would need a cell cycle that lasts 45 minutes, which we don't have. So basically, what we conclude from that is that there's something else going on. Either there's some memory mechanism or some averaging mechanism. So averaging has been proposed, and right now I personally think that that is the most likely thing that is going on. Also remember that our k-on, k-off measurements are effective, so they should take into account some elements of this, but probably not all of the averaging elements, because some of that does happen at the mRNA level that then gets shuffled between nuclei, which we don't have. So, but we were interested in asking the question of, is there memory? And so we basically looked at the probability that you express, you're active given that your mother is active, or you're inactive if your mother is inactive, versus the other way around. And we looked at the whole embryo at the border, and we compared the sample. So these are the ones that are active daughters from active mothers and non-active daughters from non-active mothers compared to a random sample. And then we did it also for triplets, 2 daughters of one mother and so on. And basically, this is sort of the best embryo we have, but I would say it's unconclusive. So we're still trying to investigate that. Okay. So actually, I have like 5 minutes left. Okay. So yeah, so the fly, I mean, part of the interest, as said, in the fly is its precision, and a lot of ideas have been put forward about information transmission. It's been shown that to be a system that transmits information fairly well. So sort of inspired by all these problems of time, actually, how should I do this? We were interested in asking what kind of network is good for making a delayed readout. So if you have some input, say some sugar or some input to a receptor in a two-state signaling system, and you have an output, but you make the readout of this output at a later time. What is the kind of network that best optimizes the information between the input at a given time here and the output at a later time? And so you can write down the information for it, and basically to make things simpler, we consider the system where both the input and output are binary variables, so they can either be on or off. And we fixed a characteristic timescale for the system at which you make the readout in terms of the relaxation time of the system. So one of the things we found is that the best way for these kinds of systems to actually transmit information, assuming that if they're in steady state, is to use the fact that you can have slow and fast timescales. So the whole system is initially in steady state, and you break detail balance, you induce a rotational order, so you cycle through the states, but some of these transitions are fast, that's the red ones, and some of them are slow, are these. So this basically helps you figure out you're most likely to be in these states, because once you end up here very fast, you'll go here in a very fast way, and this helps you figure out where you're coming from. And the topology of these systems corresponds to these sort of push-pull networks. Okay. But if you allow the system to be out of steady state, then a different solution appears. You still use the fast and slow timescales, but now you have a solution with absorbing states. So your system is most likely to initially be in either this end state from which it cannot expect, which is very informative, although pretty useless, or in this state, in which case it has to go through this specific, this whole cycle, and knowing, again, with this fast and slow time scale, you pretty much know where you're coming from. And so this is a sort of a situation where you kick the system out of steady state, for example, by supplying a sugar, then quickly you start metabolizing the sugar. It takes some time. When you're done, you go into a dormant state. And so again, you're using this slow and fast time scale, but you're mainly using the fact that you have very limited probability to be in the two systems to two states to know how much information you're transmitting. And these kinds of push-pull networks are actually very common in stress response when you are kicked out of some sort of comfortable state and have to relax to it. But the thing, and I'm not going to have time to explain this, but when you look at those systems, the thing that strikes you is that they're all out of equilibrium. So we were wondering whether we can actually calculate the entropy dissipation associated with it, so I won't go through it, but you can basically, just from defining the dissipation for a given network as the probability of going along a cycle in one direction versus going in the opposite direction, you can reduce it to a more tractable formula in general, even when you're out of steady state, which in steady state reduces to a formula you're probably more familiar with. And the point is that the circuits I discussed previously, they all have infinite dissipation, even when they're in steady state. So we started investigating what happens if you actually impose some constraints on how much dissipation you can transmit. And what happens is that if you have less dissipation at your disposal, then of course you can transmit less information. And so in the simplest case, when there is no feedback, all you can do is play around with the rates. But if you do have feedback, a solution appears, and despite of how much dissipation you have at your disposal, except when you really have zero, when you're zero, you're dead. You can't do anything. But other than that, you can transmit a lot of information despite your dissipation constraints just by using this fast and slow timescales that help you align and anti-align the system. And the interesting thing is that when you're in the infinite dissipation regime, which we completely out of equilibrium, which I showed you before, you have this negative feedback. But in the slow dissipation regime, you're really in a limit where positive feedback helps you. And this is kind of akin to what we saw in continuous systems with molecular cost, that for when you don't have a lot of molecular molecules at your disposal, it's positive feedback that helps you also. Okay, so I'm just gonna stop probably. If you're interested, I mean, we actually did look at what happens calculating things for when you're out of steady state and how costly it is to how much you have to pay for when you're kicked out of steady state. You can then transmit a lot more information than when you're in steady state, but you have to pay an additional price in terms of dissipation. But it turns out that this price is not so large, especially if you're especially if you're sort of nothing happens later for a very long time. So I'll stop here.
So when you compared the precision of the theoretical model against the data, so how did you do that comparison at the level of hunchback or at the level of the occupancy of the promoter? And
no, at the so we forget. You mean we don't have, in a way. Let's say we don't have access. No, it's at the level of the fluorescence coming out from the transcribed mRNA, which we're assuming corresponds to the occupancy of the promoter.
But you can have spatial averaging at that level by exchange of mRNA between neighboring nuclei.
I doubt it, because what you're measuring is a spot, right? It's a locus, and you're measuring a spot in the nucleus. I mean, really, what we're
okay, so
you know, we're really p-on is really linked to this, right? This is a trace. It's the intensity, divided by the maximum possible intensity, right? So, and that's really what comes out of the locus. So at this level, I don't think we have.
Nuclei does not play a role.
Also, especially since what we're measuring now is this, is a transgene, so it's not. Well, that doesn't really matter. I mean, I think we're just really measuring it at the as it comes off. Then real hunchback, of course, averages, and there is some, and you know, and bicoid is self-averaging. So that's why I think in the K on and K off we're measuring, it is an effective rate that does account for all this averaging. So I don't think, you know, we're completely average free, but at the level of the output, I think we are average free. But I'm, you know, I get confused
Yeah.
in, so.
What goes in there?
Last question?
You fit the rates with the autocorrelation. Is there a reason that's, so you know the likelihood function for a two-state markup process?
Yeah.
What about the autocorrelation is better or worse than just maximum
Well,
likelihood?
it's less noisy. It's as simple as that. I mean, we're already dealing with a lot of noise. So initially what we wanted to do, and we're still thinking about doing it, is basically doing maximum likelihood inference. But the autocorrelation function has the major plus that it's a summary statistic, so it's just the signal is much more reliable. And already with this, as I said, real data, real world problems, it's not great, right? So now we're a bit more skeptical about doing the maximum likelihood. I think there's, you have to calibrate the signal much better. I mean, we are trying to do that. We're also trying to get more and more embryos to get more and more statistics, and deal with that. But I mean, I would say that the plus of the autocorrelation function over maximum likelihood is that it's just you don't deal with the noise that much.
Thank you very much.